In this video, we're going to look at how we can repair DNA through homologous recombination. And up to this point, we never really even talked about why we would do homologous recombination. And we saw that prokaryotes have the proteins to do homologous recombination, but they don't have another chromosome. So why can they do it? That's because the vast majority of homologous recombination is used to repair damaged replication fork. And this happens roughly once a generation for E. coli and maybe 10 times per eukaryotic cell cycle where your replication fork gets stalled. And using homologous recombination for this purpose is called recombination repair. And doing recombination repair can lead to changes in your DNA. So it's highly mutagenic, last resort way to fix your DNA. And RUV-A and RUV-B, which will take care of homologous recombination for us, are turned on by the SOS genes like we talked about. So this is really a way of, there's no other way to repair this DNA. So let's try homologous recombination. So let's go through the steps of this. So let's say you are repairing DNA. So here we have the leading strand in blue and the lagging strand in red. Let's say you come across some kind of nick or gap where you have not repaired this DNA damage yet. So what will happen when you hit this gap is that your fork will collapse. The replication machinery will dissociate and you can no longer continue replicating. So we're left with something like this. Our leading strand has been stopped because we couldn't get past it, but we do have a template of our lagging strand. So even though this is colored dark blue, this hasn't come, we haven't done recombination yet, so don't be tricked by that. This dark blue is just saying we have a template for filling in our dark blue chromosome here. So what happens is that rec B, C, D, and A will bind like we talked about in the previous videos, and we will have strand invasion. So our lagging strand will go and invade our leading strand here, and so we can continue synthesis of our leading strand. Then we will have a holiday junction form. So where the gap was, where we stopped, that can go and invade our leading strand DNA, and now you can see we have formed our holiday junction, and this is carried out by RUV-A and B like we talked about. Once we have our holiday junction, RUV-C will come in, and it will cut our holiday junction, and now we have successfully gone over our gap, which is shown with this red DNA, and we can continue replication. And the way that we continue it is basically we treat the ending strand of the nick as an Okazaki fragment, and so we have machinery to fix Okazaki fragments. So we have no problem restarting up this process. Now to restart the DNA replication, we need a different mechanism, because remember we need an origin in prokaryotes to start up replication at the OREC. Well, we can't do that here. We're not at the OREC. So there's a whole new other mechanism, origin independent replication, that can be used to restart this, and it's carried out by the primosome. So the restart primosome will come in and it will restart our replication fork. This 5' end is going to be treated like an Okazaki fragment, and we can just start DNA replication from there. Now we can also use this process to fix a double-stranded break. So previously it was just a single-stranded, so let's look at a double-stranded break. So the first thing we need to do is we need to chop off the ends of our double-stranded breaks to have overhangs like this, to have ends that are now single-stranded. Then rad 51 can come and we're going to form our holiday junctions. So 1 3' end will go and invade our neighboring chromosome or our homologous chromosome, and our other 3' end is going to interact with the strand that has been displaced. So once we have our templates ready to go now, polymerase will come in and fill the gaps. So that's what these more light colored lines are showing. They're showing that the new DNA polymerase has come in because they have a template now, and we're filling in our DNA with the information we were missing from our double strand break. And we are creating holiday junctions here. So we have 2 holiday junctions from doing this process. Now we need to resolve these genes and are these holiday junctions, and when we do, we have our double-stranded DNA that did have a break here, and now you can see it has been fixed. One of the gaps has been filled in with our other chromosome here in blue, and our other gap is filled in through our polymerase action. Now rad51, the enzyme that is responsible for doing this, will interact with both BRCA1 and BRCA2 during this process. And we know that a loss of BRCA1 or BRCA2 shows a large increase in breast, ovarian, or prostate and pancreatic cancers, up to 80% lifetime risk increase if you lose these proteins. So you can see that these proteins are very important for making sure that when you have a double-stranded break that we fix these correctly. If we don't, we lead to mutations, which can lead to cancerous cells. That ends this video, and I will see you in the next video.